and I will be presenting immunoinformatic strategies on a computationally designed vaccine derived from the epitopes of the Zaire Ebolavirus glycoprotein. So there are two things I want to establish about this disease. Number one, the virus is severely lethal. So its mortality rate has peaked at 89%, killing more than half of the people that get infected. Number two, it is also extremely contagious. In fact, it is the most pathogenic strain of the five that are known to exist. Regarding the transmission of the virus in severely infected patients, less than 1% are at risk for those who haven't been exposed to sick patients, 2% when you live in the same household, 23% when you've had direct exposure already, and 50% when you are the caretaker of the patient. Finally, 83% for those who handle the corpses of those who were sick become at risk for getting the infection. So these are people who get rid of the body or perform burial rites. So the structure of the Ebola virus comprises of 7 genes, but out of the 7, the glycoprotein is what is usually selected in the making of antiviral compounds. So the glycoprotein is also the only protein that is expressed on the surface of the virus, appearing as spikes. So this protein is involved in viral entry onto a host cell through attachment and membrane fusion. So additionally, the Ebola virus has a strong binding affinity to antigen-presenting cells, which in turn leads to massive recruitments of other dendritic cells and macrophages. So targeting the glycoprotein can inhibit viral entry and membrane fusion, as well as induce the innate and acquired immune responses because of its high binding affinity to MHC class molecules. So the objectives are as follows. To predict, assess, and filter the CTL, HTL, and LBL epitopes of the vaccine, to select the epitopes and HLA combinations predicted to be recognized by West Africa and the world, to employ an appropriate adjuvant and to select for specific linkers for vaccine assembly, and to simulate and interpret the immunological profile induced by an injection of the vaccine. So for the methodology, number one, NCBI will be used for retrieving the FASTA sequence of the targeted protein. NetCTL will be used for CTL prediction. IEDB will be used for HTL prediction. And IBCEEL will be used for LBL prediction. So their assessment is done by checking first the immunogenicity for CTL epitopes, toxicity, allergenicity, and antigenicity. And finally, cytokine inducibility for HTL epitopes. So epitopes that pass the parameters are then partnered with MHC1 and 2 alleles. So for here, multiple sequence alignment will be done to account for mutations. Population coverage of the CTL and HTL epitopes is done to determine the global coverage of the vaccine construct. And finally, assembling the vaccine would include selecting an appropriate adjuvant and specific linkers to string the constituents together. So the assessment of the fully put together vaccine will be tested again using the same parameters from the filtering stage. And its physical chemical parameters are also identified to validate the stability of the sequence. And for our last step, the in silico simulation is executed to see how long the vaccine would last, how high the activity is, and how protective the vaccine construct is in a span of 1 year. Over here, table 1 shows the accession number and other details regarding the FASTA sequence of the Zaire Ebolavirus glycoprotein. So I got this from Uniprot, and all succeeding experiments will come from this sequence. So displayed in this slide is the process of filtering the CTL epitopes. So the image only shows a chunk of the amount of peptide sequences that were filtered. For the results, all peptide sequences arranged by immunogenicity in descending order have been filtered out to be immunogenic, non-toxic, non-allergenic, and are antigenic above a 0.8 threshold. So initially I had 213 epitopes, but through filtering, I was left with only 10 candidate epitopes. For here naman, for the MHC alleles, all allelic partners have been filtered out to have a percentile rank score of 1.00 or below, since a low percentile rank score suggests high binding affinity of the alleles to the CTL epitopes, which is what we want to have successful antigen presentation. So for the CTL epitopes, for the 10 of them, I was able to identify 57 MHC alleles to partner with. This is good because an ideal epitope should have many allelic partners, because this suggests a wider reach for recognition. So for HTL, IEDB again was able to predict 51 combinations under a 0.25 percentile rank threshold. So after filtering, I was only able to obtain one peptide that was able to induce all 3 cytokines needed in the study. So these are IF and gamma, IL-4, and IL-10. For IFN gamma, one of its main functions is to interfere with the infection process of a pathogen. So its secretion boosts the activation of macrophages and induces MHC molecules for further antigen presentation and recognition. IL-4, on the other hand, acts as a mitogen for B cells. So it can influence the production of antibodies by helping B cells mature and differentiate. IL-10, this cytokine prevents tissue damage by suppressing the upregulation of pro-inflammatory properties induced by an overwhelming response of the adaptive immunity. For LBL epitopes, the prediction, I was able to predict 174 epitopes, but after filtering, I was only left with 5 candidate LBL epitopes. So after that is multiple sequence alignment. So the figures on display here are the results of the multiple sequence alignment viewed with the MView server. So very minimal mutations were identified, and the epitopes of my study were highlighted in yellow, as you can see on the screen. So I was able to count how many conserved epitopes there were in the 533 sequences aligned. So almost all similarity scores exceed 96%. So this means the selected epitopes would be able to induce CD8 T cells, CD4 T cells, and B cells, regardless of where the infected may catch the virus from, since the sequences were randomly taken at different areas. So this means the chosen epitopes would allow for a widely distributed vaccine in terms of strain mutation, making the design persistent and efficacious. So for population coverage naman, we do this to locate the geographical area in the world with the highest frequencies of alleles that match the epitope and HLA combinations found in our vaccine. So globally, the vaccine is predicted to cover 100%. What this means is that majority of the global population can recognize the epitope and HLA combinations in the vaccine. Now, the epicenter of the Ebola outbreak occurred in countries from West Africa. So I also had to highlight the score here. West Africa achieves a coverage of 99.18%, making the vaccine construct reliable to those more in risk of the infection. For the vaccine construct assembly, the adjuvant that I picked was CD40L. So this actually is a co-stimulatory molecule involved in the activation of helper T cells and B cells, since it's involved in the success of antigen presentation. So total activation of the helper T cells is the result of a thorough interaction between the CD40 molecule of antigen-presenting cells, such as B cells or dendritic cells, and the CD40 ligand of the T cell. So this can positively influence the differentiation of B cells. For vaccine assembly again, this is just a representation of what I have from here. EAAK is a rigid spacer whose function is to ensure minimal interference between domains by providing distance in between the epitopes and the other elements in the vaccine. So I use this to connect the adjuvant to our CTL epitope. AAY spacer naman, this spacer serves as the proteasomal cleavage in sites of cells of mammals. So peptides connected by AAY are not challenged by separation difficulties. So this can increase the immunogenicity of individual CTL peptides. GPGPG, this is designed to be a universal spacer. So this linker is able to induce responses of helper T cells. And finally, KK is the last linker I used. I use this for LBL epitopes. Using this linker prevents the induction of antibodies from joined junctional peptide sequences. And finally, I have my polyhistine tag, which is just used for purification purposes. So the properties of the now strong construct, the vaccine construct, since I tested again with filtering parameters, this includes the retention of its antigenicity, allergenicity, and toxicity. So they all still pass. And the physicochemical properties, they show good stability of the vaccine, which is indicated with the instability index. It's below 40, so this means that it's still stable. For my CMSIM results for cytotoxic T cells, the graph on the rightmost one, shows the population of active CD8 T cells. So you can see that it has a heightened trend for the first 5 months after injection. So the active CD8 T cells decline months after injection since repeated exposure to the antigen is no longer induced. I want to say that again that I only had 3 injections. So after a few months, the trend goes down. So this just means less antigen presentation, therefore less activity from CD8 T cells. On the other hand, resting CD8 T cells goes up naman after injection, which amounts, it has the same amounts by the end of the year, correlating to the population of CD8 T cells on the left graph. For the graph on the left, although it shows no memory CD8 T cells were formed, their population was still abundant months after injection. So this suggests that the CTL epitopes that I predicted were able to repeatedly induce CD8 T cells. This indicates a strong immune system ready to respond to infected cells should exposure arise. In addition to this slide, no CD8 T cells were found to be anergic, which meant a successful interaction between the cytotoxic T cells, the targeted cells, and macrophages, signifying a responsive immune system. For helper T cells and cytokine secretion, excuse me, the graph on the left shows the population of CD4 T cells. So besides memory B cells, CD4 memory cells also contribute to the longevity of a vaccine-induced immune response. So for the total population, we see the black line, we see a steep decline after 3 months. This is because most activated antigen-specific cells don't survive the immune response against the first exposure after activation. So now we also see a trend for memory CD4 cells, which is the light blue one, declining in a slower pace naman after 3 months. So the memory CD4 T cells that we have are actually the result of surviving cells. So now they outnumber naive CD4 T cells because they proliferated, which allows them to detect the same antigen faster. So this graph also shows that we have memory helper T cells still present by the end of the year after injection. The graph on the rightmost naman shows the 11 best responding cytokines induced by our helper T cells. So again, we have IFN gamma, which is very abundant. IFN gamma is the purple peaks that we see, and it peaks only 3 times because it was secreted in response to the antigens present in the injections. So we don't see it linger because it was really only there to prevent the spread of the infection process. Our second cytokine that we made sure to was induced is IL-10. So IL-10 limits tissue damage caused by overwhelming immune responses. So it wasn't shown to be largely present. So because its induction was really small scale, we can assume that the vaccine construct is not overpowering and is well received by the body. Finally, we have a smaller graph that you can see on the right side of the right graph. This graph is for interleukins 2 and danger signals. IL-2 is the yellow peaks. So IL-2 is responsible for promoting regulatory T cells. Now, because that concentration of IL-2 is really high compared to the danger cells, it indicates that a sufficient amount of T cells were produced in response to the antigen. So danger signals are low because the amount of T cells produced are enough to overcome an infection. Finally, for B cells, plasma B cells, and antibody production, the graph on the top left shows our B cell population. So there's a trend here that shows a strong presence of long-lasting memory B cells. It's the green line that you can see that has a big slope. So the strong presence of long-lasting memory B cells, as well as the repeated induction of mature class switching B cells. You can see that there are different isotypes of B cells. So what this means is that there's a diverse humoral immunity, which is equipped and ready to defend the body. So the state of whether these B cells are active, internalizing, presenting, duplicating, or anergic is depicted on the graph beside it on the top right graph. So on the top right graph, what's important to see here is that active B cells are still steadily present 2 months after injection until the end of the year. So there's a little blue line in the points of injection, the 3 points of injection here that shows the amount of duplicating B cells. I think it reaches only around 100 cells per cubic millimeter, so we can correlate this to the graph on the bottom left. So the graph on the bottom left is for plasma B cells. So the duplicating B cells are meant to be in their division stage. So this means that they differentiated into plasma B cells. So here we see that plasma B cells are present during days close to the injection points, but die down pretty fast. So this is because plasma B cells are only induced in response to antigens. So they no longer need to be induced once there's no antigens present anymore. So these plasma B cells also secrete antibodies seen on the bottom right graph. So because one plasma B cell can produce hundreds of thousands of antibodies, antibody levels are shown to be very concentrated during injection and are less concentrated after. Finally, for my conclusions, which I placed beside my objectives, epitope prediction yielded 10 CTL, 1 HTL, and 5 LBL epitopes. 57 MHC allelic partners were identified, and a vaccine coverage for West Africa was achieved to be 99.18%. Globally, this achieved 100%. For here, CD40L was chosen as my adjuvant. EAAK, AAY, GPGPG, and KK were chosen as linkers. And finally, An induction of CD8 T cells, CD4 T cells, and B cells continued months after injection, showing immunity.